that's in the muscles. So the glycolytic system on slide 29, there are pros and cons. The cons is that there's a relatively low ATP yield compared to the slow glycolysis that we're going to talk about next. And not only that, but if I have a lack of oxygen in my system, and I'm not breathing, like when you're swimming underwater, you're not breathing oxygen. You just have oxygen in your system. But if I do have a lack of oxygen in the system because I'm holding my breath, or because I just don't have oxygen flowing. Maybe my iron content is low, so I don't have a high hemoglobin count, so I'm not processing oxygen. There is a better increased chance that you're going to create a lactic acid. Lactic acid impairs the breakdown of sugar, and it also impairs muscle contraction. So there are cons, but the pros are that this particular glycolytic system, the fast glycolysis anaerobic glycolytic system that leads to pyruvate and lactic acid, this particular system allows muscles to contract when there's no oxygen. So at least you can swim underwater and like maybe if you have to hold your breath, you'll be able to move around and at least conduct activity. And not only that, but it does actually create more ATP than the phosphocreatine system, so that's a pro. And it also actually, the breakdown of the sugars through the glycolytic system provides a little bit more higher intensity explosion, or higher in intensity energy availability. So it's actually better for higher intensity activities. It's just that the metabolism, you can burn more energy another way, and the other way is through the oxidative system. And we're on slide 31 right now. The oxidative system is aerobic. It utilizes oxygen. That's a great thing because there's oxygen everywhere. So this like biochemical being has evolved, and back in the day when it was like this little molecule in the water, it started to realize, wow, if I could just use the oxygen above the water in the air, I could start to make more ATP. Now you look over here, there's 32 to 33 molecules of ATP for 1 molecule of glucose. So you get like a 30-time increase in the amount of ATP that you can utilize if you use sugar and you have oxygen, and that's the key, all right? So you have a much longer, steady supply of oxygen. It's everywhere. You can't get away from it. And oxygen is probably, the highest physiological need. Oxygen and water are the two highest physiological needs that your body must have in order to live. And all of these processes are now going to occur in the mitochondria if there's enough oxygen present. So the glucose molecules are going to be, or the carbohydrate molecules are ripped apart. Those are created, pyruvates are created, and those pyruvates are going to combine with oxygen, go across the membrane of the mitochondria, and start to go through what's called the Krebs cycle. The Krebs cycle is where a huge number of processes started by acetylcholine and citric acid, those are going to combine. They're going to go through a bunch of processes. They're going to release ATP all along the way. A lot of those processes are going to result in like electrons and these things called FADH, and those are going to go through what's called this electron transport cycle. It's going to break down all of those into even more ATP. Bam, and then you get 30 times more ATP. So that's a pretty good thing. I can't go through all the processes of all of the we're on slide 32. I cannot go through all the processes of glycolysis, Krebs cycle, and the electron transport chain. I'm on slide 33, figure 2.8. I can't necessarily explain every single step, but what it's saying is at the top you have glucose. Glucose is going to get broken down. And that's going to result in the loss of 2 ATPs on the first step and 4 ATPs on the second step. And then those glucose molecules are going to make 2 pyruvate molecules. And those pyruvate molecules are going to combine, and they're going to break down, and eventually they're going to release carbon dioxide. That's the 2CO2 there. And that happens inside the mitochondria. There's a release of carbon dioxide. That's the carbon dioxide that gets in your bloodstream that you breathe out. And then eventually those 2 pyruvates are going to be broken down into 2 acetyl-CoA molecules that are able to go through that Krebs cycle. And the first step is going to result in a release of those 6 NAD and NADH. Those are ions. Those have electrical charges, and those electrical ions are going to be brought to a electron transport cycle. I can't explain everything, and everything results in, look at the bottom there, a result in 28 ATP. Holy smokes, that's what we like. All right, so glycolysis, if you go, we're on slide 34, glycolysis can occur with or without oxygen, but with oxygen, it really creates much more ATP. It takes a long time to do that. That's why it's called slow glycolysis. It takes a lot longer to go through the Krebs cycle and make ATP through the Krebs cycle, but you get a lot more ATP, and you don't have to really search for the resources. It uses oxygen. It's in the air. It's free for us all. Right now, I'm on slide 35. One molecule of glucose leads to two acetyl-CoA molecules, and those acetyl-CoA molecules are the molecules that are going to be able to go through the Krebs cycle. I can't go through the Krebs cycle process like the book goes through. I'm not a biomechanist. All I know is that eventually there's going to be those hydrogen plus molecules. Those hydrogen plus molecules are going to go through this process that's on page 36. That is the Krebs cycle already. I can explain that to you. Figure 2.9. The pyruvate is going to combine with NADH, and it's going to result in NAD plus, and it's going to combine with that to release carbon dioxide and NADH and create acetyl-CoA. The acetyl-CoA is going to combine with that CoA, coenzyme A, and that coenzyme A is going to result in a change of the acetyl-CoA to citrate. And that's why they call it the citric acid cycle, because citrate is an acid. That's going to combine with ankinase and create this new molecule called isocitrate, which is going to combine with isocitrate dehydrogenase, and that's going to result in a release of positive and negative ion charged molecules called NAD and NADH. That's going to also result in a release of carbon dioxide. That's going to result in a storage of those ions, and those ions, positive and negative charged molecules, are going to eventually go through the electron transport chain. On slide 37, it talks a little bit more about the electron transport chain. I can't really talk too much about this aside from the fact that it creates a huge amount of energy. So if you go to slide 38, it talks about how much energy one glucose eventually is going to break down and make 32 ATP molecules. So one amount of sugar equals a lot of ATP. And that's why your body uses sugar, because you can create a lot of ATP. That's why we like sugar. That's why we like pasta and potatoes and carbohydrates and Twinkies. All right, well, this talks about how this is like, it shows figure 2.1 talks about the mitochondria. It shows the processes by which the sugars are going to go through the mitochondrial membrane and get into the mitochondria so that it can start to break down into ATP. Again, I don't think that right now this is at the scope where you guys need to go. I think it's more complicated than you need, and I think that if you're interested in the biochemical processes, then I think that you should seek out more information regarding the biochemical properties. But having said that, me being able to do this lecture right now gives me the opportunity to delve a little bit further into the biochemical processes, and maybe I can deliver that stuff in lecture as opposed to doing the obligatory lectures. So I think this frees me up, this kind of lecturing here frees me up to find information that I don't know that might be more difficult for you and present that in class as opposed to all the other stuff that I think that you can get on your own. All right, so now we have figure 2.11 on slide 40. It just is another summary of everything. You have glucose, the top left. Glucose is going to combine with 2 ATPs to make 4 ATPs, and eventually pyruvate at the bottom. That pyruvate is either going to go through the fast glycolysis process and get broken down into lactic acid and result in a small amount of ATP, or it's going to keep going with oxygen. It's going to combine, and it's going to become 2 acetyl-CoA molecules and go through the Krebs cycle. That Krebs cycle is going to take all of those ions, those positive and negative charged molecules that are released in each step. It's going to shuttle it to the electron transport chain, and it's going to make, boom, 5 and 5 and 15, and another 3 and another 2, and totally it's going to make 32 ATP molecules for every molecule of glucose. We're on slide 42 right now. When you break down fat, fats start as triglycerides. You have 3 fatty acids, and those fatty acids are combined to a glycerol molecule. And through a process called lipolysis, lipolysis, the breakdown of fat, you can get energy. The 3 fatty acids that are released through your lipolysis are going to eventually make more ATP than glucose. But it takes much more time for fat to break down than glucose, but I mean, you do get more energy. You cannot use those free fatty acids for energy directly. They have to go through what's called beta oxidation on slide 43. Eventually, because you remember, carbohydrate and fats are all made of carbon, hydrogen, and oxygen. So all of those particular molecules, both carbon, fat, and eventually protein, are going to eventually break down into the same molecule, which is acetyl-CoA, and that acetyl-CoA is going to go through the Krebs cycle. Okay, so in terms of slide 44, eventually, like we said, the breakdown of fat, acetyl-CoA, is going to get into the Krebs cycle, and from there, it's the exact same process of glucose oxidation. Depending upon the number of carbons are on the free fatty acids will determine the amount of or the number of acetyl-CoA molecules, and therefore the amount of energy that you can get from that particular fat. On table 2.2, it just shows the amount of I believe ATP produced from one fatty acid. And in this situation, as opposed to one molecule of glucose yielding 32 molecules of fat, calories of fat, molecules of ATP. Here, one fatty acid from palmitic acid yields 106 ATP molecules. So you can see the ATP production of fat is much higher than that of glucose. Protein, we're on slide 46. Protein isn't really used as a substrate because it takes energy to break down protein. It says on on that slide 46. Nitrogen on the molecule makes it into a pretty unique process because you have to spend energy to break down the nitrogen group to get energy. So your body doesn't like to spend energy to get energy. All right, the Patriots are starting, and I will do the rest of this chapter in a bit.